I know it's going to be a very long lecture, but we have to complete what H1N1 and the story of H1N1 is much more interesting. So in April 2009, the WHO received reports of sustained person-to-person transmission with a new influenza H1N1 virus in Mexico and USA. Always so now remember that everything doesn't start in China. This has started in Mexico and United States. The virus currently affecting all regions and which are pandemic alert states, sporadic outbreak in field. So H1N1 is related to pig. If we see the timeline of H1N1, the first outbreak has happened in Mexico of a pneumonia-like illness. In April 15 to 17, there is notification of clustering of rapidly progressive pneumonia cases. That is why a typical pneumonia presentation, which is not recovering to a usual line of treatments, should be reported to WHO, higher authority WHO. A case of atypical pneumonia happened in Oklahoma State. It provided to enhance surveillance. April 15 to 17th, two cases of new H1N1 virus infection identified into Southern California. Case in USA, April 23, you have more infections coming up in Mexico. After 26 April, it is spreading to Canada, then it is spreading to UK, US, New Zealand, and in 27th April, WHO declared it as phase 4. So phase 4, I told you about what is phase 4. That is human to human transmission, and phase 5 is bigger transmission than that. But the most important declared that is June 11th. It came into control in June 11th when the WHO declared the phase 6 of the pandemic, and it got controlled within 1 year. But most importantly, it is not got controlled. You can see that it has every year we have cases of H1N1 which do not get which get reported, but we do not come to know that they are there. I will show you that. Northern Hemisphere. In the Northern Hemisphere, there's an increasing trend in ILA cases. What is ILA? Influenza like illness. It's been reported in most temperate countries. So those countries which are over 30 degrees latitude, they will report influenza like illness. So with countries like USA, France, USA, Northern Ireland confirmed patients with H1N1 positive. And those southern hemisphere were below the baseline in the temperate countries have reported cases. So this is the pandemic H1N1. So I told you about H5N1, H1. This is H1N1. Okay. So the pandemic is this called the pandemic. What is the pandemic? A pandemic which affects larger areas, geographical areas, or spread through more continents. This is a pandemic where the majority of the pandemic you can see more cases are in the United States, Mexico, United States, South America, spreading to Europe, Australia, and I went to China and India also. So I had a work in, I had experience that I was in first year residency, second year residency in community medicine during the pandemic, and we were posted to screen patients the symptoms of H1N1, and then they screen them for a probable case or suspected case and then admit them. The problem statement: 11 to 21% of the global population, or 700 million people, contracted illness more in absolute terms than in Spanish flu. Actual fatality rates were between 12,000 to 18,000. The CDC estimates that there were much more fatalities. Around 2 lakh people have died, as the CDC tells. In August 2010, the WHO declared that the pandemic is officially over. That is 1 year after it started. But we'll. So what are the aging
Antigenic shift
factors?
and the
Yeah. So you will
H1N1
see this video.
antigenic shift and the H1N1 influenza A virus. Antigenic shift occurs when the influenza virus goes through the major antigenic changes as a result of the reassortment of genomes among different strains, including animals. Origins of the hemagglutinin HA, nucleoprotein NP, and non-structural NS gene segments of the H1N1 virus are linked to avian flu virus, which infected pig populations around 1918. Let's review the principles of antigenic shift. Influenza sets up local upper respiratory tract infections in pigs. The virus targets and infects epithelial respiratory mucosa cells. Hybrid viruses are created when they infect the same cell by different strains of the virus. First, the native pig strain of influenza A targets the epithelial cell. It binds and enters the host cell through endocytosis and releases its viral genome into the cytoplasm. Secondly, a bird strain of influenza virus A infects the same cell and mixes with the already present pig strain gene segments. Mixed viral gene segments from bird and pig strains reassort into a new influenza A virus strain, which may further spread to the human population. Another major antigenic shift of influenza A took place around 1979. The neuraminidase or NA and matrix MA gene segments originated from wholly avian influenza viruses and entered the swine population. Substantial antigenic shift of viruses have occurred since 1979. For example, when the polymerase acidic protein or PA and polymerase basic protein 2 PB2 segments from the bird strain reassorted into a new strain, which entered swine in North America around 1998. Around the same time, the polymerase basic protein 1 PB1 gene segment migrated to a swine strain from humans. This had originated from birds around 1968. Evolution via the combination of various antigenic shifts resulted in the novel H1N1 transmitted to humans in 2009.
So this was so I could explain it. This was a very good explanation. It's a very good YouTube site. You can go and see a lot of mechanisms in medicine are shown there with interactive animated medical education. So as you told, the reservoir of infections are usually the avians and the pig swine population where it is on. So you can see from 1950s what were all the reservoirs. Pigs are the most important reservoir for influenza A virus, which acts as the intermediate host for both the interspecies transmission and the genetic risk. The case definitions and the probable case definition, you should know the probable case definition is any acute febrile illness which is positive for influenza A by influenza RT-PCR is positive for influenza A by influenza rapid immunoassay is an individual who is in suscept is a clinical compatible case who died of an unexplained acute respiratory syndrome. That's a probable case. But a suspected case is the one who is an acute febrile illness more than 30 degrees Celsius. Within 7 days of close contact with a person who is a confirmed case of H1N1, within 7 days of travel to areas where there are one or more confirmed cases or resides in a community where there are one or more cases of H1 influenza. So the confirmed case, a confirmed case of a pandemic is one which is proved by a real-time RT-PCR, a viral culture, and a fold-fold rise in viral influenza virus-specific neutralizing antibodies. Okay, should know this is what is called as a confirmed case. Incubation periods is up to 2 to 3 days. It could range from 7 days. Symptoms are mostly of non-febrile, can be ranging from non-febrile mild upper respiratory illness to febrile illness, influenza-like illness, or severe or even fatal complications, including rapidly progressive acute respiratory syndrome. Complicated severe influenza, it can lead to lower respiratory disease, pneumonia, encephalopathy, encephalitis, or septic meningitis, renal failure, multi-organ failure, abdominosis and myocarditis, and exacerbation of underlying chronic diseases, including asthma. Treatment. Uh, so the treatment usually you will collection of the nasopharyngeal samples and the throat swabs are remember for that thing. This is a video will show
COVID-19
for
diagnostics.
diagnostics, which is
Performing a
COVID-19, which
nasopharyngeal
we
or
can use it to
oropharyngeal
show
swab.
how the collection of the
It is
nasopharyngeal swab
essential
is
to
done.
follow proper infection control measures when samples are collected from patients with a suspected coronavirus infection. Among other things, the examiner should wear an N95 respirator mask, gown, protective goggles, and gloves. To save resources, we will refrain from using each of these items in this video and instead only show the technical details of sample collection. Let's begin with the nasopharyngeal swab. The patient should be alert and cooperative. Allow them to first blow their nose. The patient should sit in a relaxed and comfortable position during the sample collection process. Tilt the patient's head back slightly and support it with your non-dominant hand. If necessary, lean the patient's head against a wall to minimize jerky movements. Stand slightly offset from the patient to avoid the risk of contamination in case of a sudden cough or sneeze. Be sure to advise the patient of potential discomfort during sample collection and let the patient know that they are welcome to close their eyes. Sample collection should only be completed using sterile swabs made from artificial cotton with flexible plastic shafts. Calcium alginate swabs or swabs with wooden shafts must not be used. These materials can inactivate viral particles or inhibit PCR tests. Furthermore, wooden shafts are more likely to cause patient injury. Hold the swab like a pen between the thumb, index, and middle fingers. Maintain a loose grip that allows the swab to accommodate any resistance encountered during sample collection to reduce the risk of injury. Avoid using the following hand position during sample collection. Start by inserting the swab horizontally into the left or right nostril. Carefully advance the swab while maintaining a course that is close to both the septum and the floor of the nose, parallel to the palate. Do not advance the swab upwards, but instead straight back until resistance is felt as the swab reaches the posterior nasopharynx. When collecting samples from an adult patient, this corresponds to a travel distance of approximately 5 to 6 centimeters or 2 inches. Leave the swab at this depth for a few seconds while gently wiping the wall by twisting the swab shaft. Slowly and gently remove the swab. Immediately insert the swab into the sample collection tube, which may contain 2 to 3 milliliters of viral transport media. If a nasopharyngeal swab cannot be collected for any reason, an oropharyngeal sample can be also collected through the mouth. Again, inform the patient that the smear will feel uncomfortable for a short time and may trigger a gag reflex. Ask the patient to open their mouth wide, protrude their tongue forward, and say "ah." Depress the patient's tongue using a tongue depressor. Gently insert the swab into the oral cavity without touching the lips, teeth, tongue, hard palate, or soft palate. These surfaces should be avoided as they are often colonized by a wide range of microbes. Collect the sample from the back wall of the throat. Twist the swab shaft gently but thoroughly, sampling the oropharyngeal wall. Just as with the nasopharyngeal swab, the oropharyngeal swab should now be placed into the sample collection tube and sent to the laboratory as quickly as possible.
So this is also a very good method of collection. I think you should know about this. Students, you should know for collection of sample.
COVID-19